And
There we go. Thank you, everybody, for your patience. I think I thanked you already, everybody's. We're in business, right?
great. Folks
Sure.
on Zoom can hear me, I hope. And so I will get started. Thank you so much for having me here. I'm going to be talking about some of the robotic platforms that I build and their applications to the biological carbon pump in the ocean. And so first I'll describe a sort of low-cost camera that we built for NASA exports campaign and have since started to use in a variety of other settings, including I'm really excited to start using these in coastal systems in sort of a bottom landing capacity. And I'd love to chat more with folks that are interested in measuring carbon fluxes in the coastal area locally. I'll then talk a little bit about how that development led to the design and creation of a new type of Lagrangian float coming out of my lab at URI. And then beyond that, how building low-cost expendable floats, sort of following on one of the questions that Yui addressed last week in the seminar. What happens to these floats at the end of life if they have drifted too far away for it to be feasible to collect again? And what are the ways in which we can try to take steps to come up with creative solutions to sort of mitigate that impact? So I study the biological carbon pump. This is the process by which carbon from the atmosphere, let's see. And I can actually just use my cursor, I think here. Yeah. So atmospheric carbon is taken up by phytoplankton in the surface open ocean, represented by the green dots here. And then there's various pathways by which that organic carbon makes it into the interior ocean. One of the biggest pathways is the gravity pump or the sinking pump, by which dying or dead phytoplankton or fecal pellets begin raining out of the surface into the interior. There's also ocean mixing processes that can act to move carbon downwards, and the migratory pump or the movement, the diel migration of zooplankton and other animals that can act to move carbon vertically. And we care not only about the flux of carbon, so how much carbon is making it into the interior ocean, but also the efficiency of that export. And this matters because the depth at which the carbon reaches before it's remineralized back into inorganic forms impacts the sequestration time of that element. And so carbon that's remineralized in the upper ocean may only reside a year, a season, maybe up to 10 years. Whereas if it can get below the seasonal mixed layer, below into the sort of interior of the deep ocean or near the bottom, the residence time can be much longer, hundreds or thousands of years. And the biological carbon pump, by its name would imply it's biologically mediated carbon. It's carbon that has passed through a biological system, been transformed or fixed into organic carbon, and as such, it is highly, highly complex in terms of the ways in which not only it can be moved vertically, but also the ecosystems in which it passes through. And just to sort of highlight some of the spatial variability that you see here, this is actually based on some remote sensing and modeling work by Kelty Bisson that looked at even just the partitioning into large and small aggregate particles can be hugely variable in space and also in time. And this poses a tremendous challenge to those who try to or aim to observe this flux, because our typical or traditional methods for capturing this carbon are through what are called sediment intercepting traps. So one is shown here in this cone shape, where you basically hang this structure below the surface from a surface buoy, or you moor it to the bottom. And it collects carbon over a period of time, sinking material over a period of time, and then that carbon is collected and analyzed for its total mass and some other element compositions, but it just really represents a single location that's integrated over time. And putting these kinds of traps out are extremely expensive and laborious. And so really, I started collaborating and thinking about other ways that we could try to look at this process using different types of technology. And this was really ideas that started during my postdoc at Woods Hole with Meg Estapa, who's now at the University of Maine, and Colleen Durkin, who's now at MBARI. And I basically took the concept of the sediment trap and sort of did away with the tube and mounted a simple camera with an upward looking collection surface onto a Lagrangian float, which is what's seen here. So this is a Lagrangian float built by the Applied Physics Lab at the University of Washington. Lagrangian just means that it goes subsurface and then follows the water, similar to the BGC Argo floats that you heard about last week. And so we called this SNOWCam, and the first one that we built was deployed during the NASA Exports campaign in the subarctic Atlantic Ocean. So what's shown on the left here is a map of chlorophyll showing the trajectory of a glider in white, and then a period of very intensive operations in the little box right here, where we had a whole variety of different autonomous platforms, as well as actually 3 research vessels all converged into one area, trying to make as many measurements of the biological carbon pump and processes therein as we could to eventually be able to try to develop proxies to estimate carbon export from satellites. What I'm going to be showing you here is just a small subset of that, but it's basically the data delivered from a single sea glider that's shown on the left here that was following after the Lagrangian float that had the SNOWCam attached to it. What's on the right here is a sort of zoomed in view of that little box and shows the trajectory of the Lagrangian float, which is shown in pink as it orbited around this anticyclonic eddy, and the sea glider shown in white as it followed around after it. And so what we're trying to do by designing an experiment like this is get into what's called a Lagrangian frame, basically a water following frame, in which now we can start to minimize the effects of advection, other processes that when you just measure in a single place in what's called an Eulerian frame of view, you see variability that are driven by the advection of gradients past that location. When you're in a Lagrangian frame, when you see a change now, you are better equipped to interpret that change as a local change. And for me, I'm interested in local changes that are associated with the biogeochemical or biological processes such as productivity, mortality, and carbon export. So we also had a couple of other platforms out there. NBST and STT are both more traditional styles of sediment traps that were able to then ground truth against our SNOWCam system. This is now a time series of glider data. The top is density. We have isopycnals shown in gray and the mixed layer shown in the black dashed line. The Lagrangian float was deployed later on during the experiment for this 1-month intensive period, and you can see its drift shown in the sort of scribbly black line right there. So what it was doing was it was drifting for a period of about 12 hours, and then it would pop up, transmit its data, say its location, come back down again. And that was really how we were able to keep track of where it was going. The bottom panel here shows optical backscattering, which is a proxy in the open ocean for the amount of phytoplankton biomass and particles. And you can see it sort of higher in the upper ocean especially in the later season after the mixed layer sort of shoaled. And so one thing that I was able to do with that signal is deconvolve it into two parts, sort of a baseline part where you take almost like an average or smooth the profile and reduce some of the, like you saw in the last slide, there was a bunch of like little spackles here. You can kind of smooth that out and take that as a baseline, which is what's shown on the top. It's been gridded. And then the bottom panel is the anomaly from the baseline. So basically the original signal minus the baseline, you then have a gridded product that reflects the spikes in the data. And what really pops out now is this feature right here. And what with that feature is a lot of sinking particles. So we see a descent of that plume of about 40, 50 meters per day, which was actually very consistent with a lot of our onboard measurements of sinking flux. And what now I can, so we have this clue from this optical proxy that there was something that happened around May 16th. We saw this sort of big export flux, but it doesn't tell us what's actually there. So what types of particles are they? What size are they? You know, what was, how variable was it over shorter timescales? And so we can use the SNOWCam to now take a closer look. It's a pretty simple thing. It was cost only about $250 in parts. It has a Raspberry Pi camera that's looking upwards through this transparent lens here. It has a little LED light ring that illuminates, because typically these are dark environments that we're working in, and we need to illuminate the particles to be able to take a picture of them. And then it has some circuit boards and batteries, pressure and temperature at the bottom, and a reed switch for turning it on and off. And so what we found is that in a Lagrangian waterfalling frame, because you've really now reduced the relative flow because you're moving with the water, these particles accumulate over time on that upward looking face. And so in the top panel here, we have the drift time series of the Lagrangian float. I've indicated only the yellow times when it was actually during its drift. The rest of the time it was either up at the surface, or there was just some like reballasting weirdness that was happening in some of these spots where it was getting stuck in the mix layer. And during each of these 12-hour drifts, I got a sequence of photos that show an accumulation of particles on the camera surface. And we can now take that, take those images and segment them. We can learn something about, again, particle size, particle identity, but we can also translate it into a proxy for carbon flux. And so on the left panel here is a comparison between the SNOWCam, which is shown with the open squares, and some of our other platforms, the NBST OST and the Lagrangian float OST, that show like very, very similar patterns at high resolution. We also saw with our just comparison with our bulk traps that this was giving us a fairly reasonable proxy for carbon flux. What it adds is the ability to now see what's happening on a much shorter time scale than what we previously were able to do, and also say something about the particle types. So something that pops out from this analyzing the SNOWCam images is that we got sort of a consistent flux of small particles, but then we really started to see an increase in the flux of aggregates. And after about May 15th, May 16th, and that's really consistent with what the Sea Glider biooptical data was showing as well. And then our fecal pellet fluxes were even more variable superimposed on that. I'll highlight here some of the progress that we've made since 2021. The original raw image is shown on the right-hand side. And then this is an example of a more recent SNOWCam series of images, where the first one I was really nervous about power. And so I only had it take a picture every 4 hours. And then we realized that we actually had a lot of power and we could learn a lot from just taking a picture like every 10 minutes. And so that's what we've started to do since then. And that's an example of a recent deployment in Monterey Bay. And so if there's any questions about that, I could answer them, or I could move on to the sort of next development in our journey of Lagrangian cameras. Yeah. Yeah, it's a great question. We're working on it. We're working on it. So in that case, where it was coming up every 12 hours, that was sort of perfect because it meant that the surface got washed off periodically. And so we basically got a whole series of 12-hour durations. With the Minion floats that I'm going to be talking about right now, we don't have that yet, but we're working on it. So we're trying to actually, we're working on a little wiper that actually is the ring at the top that you saw. This we call it the diffuser ring. It basically tries to direct light toward the particles that can rotate and like push the particles off. Okay, so you know, as I guess in parallel with this idea of just the SnowCam that can attach onto a Lagrangian platform, we can also actually, by the way, attach it to anything else. You just need to add like a tube above it so that the hydrodynamic effects are minimized. I've been thinking about a way to really get into a Lagrangian frame even more than some of the profiling floats in a way that sort of allows and sort of becoming like interior ocean drifters, I guess. So trying to make something very cheap that is passively ballasted as opposed to actively ballasted, and thereby dispensing with one of the more costly elements of profiling platforms. And so this concept was hatched at a meeting in 2017 hosted by the Keck Futures Initiative. So they had an artist come and was like trying to render our discussion at the same time, which is where some of these drawings came from. And at the time I was like describing something that had like a camera looking out the side, and we were talking about its size. So they drew like a Coke can and then we're like, make it into an avocado. It was very, very optimistic. But the basic idea was that it was something that was very cheap and small, potentially a similar compressibility as seawater, that could be pre-ballasted, drift for a period of time, taking photographs, and then pop up and be expendable. And through a little bit of seed funding from that meeting and also some other federally funded and philanthropy funded opportunities, we've been able to really make these exist. And just like the sort of original rendering, they sink down to a target isopycnol, they drift for a little while, and then they drop a weight that makes them positively buoyant. And now actually the battery side becomes the heavy side. And so now they start to flip over. Inside the center shaft there's a passive traveler. That traveler then slides down to the other side and adds even more stability to the battery side, which then keeps it stable when it pops its head out of the water. And this side has the antennas and the flashers, things that we need to be able to send data and get them back. And this is really, I think, combining, just like any other like technology advancements and innovations, this really combines existing ideas together in a little bit of a different way. And so it's really adding to a family of imaging floats and borrowing from a longstanding sort of technology within physical oceanography called Rafos floats, which are much longer glass housings, but they have sort of operate on a similar principle, just measuring temperature and pressure. The Minions really aim to be affordable. So the cost of the parts is around $1,000, maybe $1,200 now. They're open source, and they're really, really Lagrangian. And this is like it is so, it's probably one of the most exciting parts for me on the science end, or on the sort of engineering side, to really be able to see and evaluate how closely these follow the water. And that is a very unique trait compared to other autonomous platforms, such as the BGC Argo floats or gliders, that will always have quite a bit of relative platform water movement. And there are some really exciting things that we can do with that. I'm just going to show this video of dropping it in the water. So they're very small, so they're easy to deploy. We've done a bunch of work on small boats, but we also work off of larger vessels. You can see initially we have to add a little bit of weight. So they sink very slowly because they're nearly neutral. We have to be very accurate with how much we trim them in order to get them to land where we want them to, rather than sinking all the way to the bottom or staying at the surface. And at first, they're slightly buoyant relative to how they're going to be once they're compressed. And so what we do to counteract that and actually initially get them to sink is we add a lifesaver candy with a little bit of extra weight. And what happens is that pulls it down, and the lifesaver dissolves after about 10-15 minutes, and the little extra weight drops off, and then the minion sort of gradually approaches its isopycnal. Sometimes that's a little bit up, sometimes that's a little down. Here's another photo of just the flipping mechanism. This was just with my tech diving down to sort of show, but it's kind of interesting how we learned a lot about how to get the distribution of weight sort of perfect as well, in terms of making sure that it had some stability even after that heavy part of the top pops out of the water. And that's where the Traveler actually is an essential part of the design. So this can also be deployed from large vessels. We did a bunch of these in exports as well, deployed by hand or with a slip line. And then when possible, we recover with a net. But they do transmit their scalar data at this point. We have not yet decided how to compress and send image information yet, but very much actively working on that. Here's another view of the Minion float taken from the ROV on board the Rachel Carson at Ambari. And so what they did was we like modified one of their sample grabbers to hold a Minion float, and then they went over the side. And then when we were at what I thought was the target depth, it would open up and kind of release the float. And then we were able to follow after it. And this was really nice because we could see the whole life cycle, including how it drops the weight, which was to burn a little, like what's called a burn wire, basically use electrolytic corrosion to release a little bit of monofilament that then drops the weight off the bottom. And here's an example of one of the deployments of a Minion in Monterey. You can see just the range of different types of particles. I picked this one because it has a lot of other stuff going on. There's like we think is a siphonophore that was sort of like swirling around in the background there. And there's also a crab that comes in. I don't know if you saw that part. Let's see. Yeah, so a pelagic crab landed. There it is in the top corner. And yeah, it spent quite a while because every one of those pictures is 10 or 15 minutes apart. So it was sitting there for quite a long time, scooping up the marine snow. And they actually captured some, and their bodies are just full of marine snow. So it's really nice to kind of get these vicarious sort of snapshots of what's actually happening. I mean, often when we deploy things, we'll get some data back and we're just like, what happened here? I mean, is this a sensor thing? Did an animal come? Is this biofouling? And so it is really exciting with a camera to begin to reconstruct what happens. I wanted to talk a little bit about Lagrangianness of the minion. 15 minutes. I'll go really quickly through this, and then we can talk a bit more. But basically, these are two examples of minions that were deployed for about 12 hours. You can see the initial sinking part. This is where the lifesaver drops, and then it sort of descends up to reach its neutral density. Same thing with this guy. One of them went a little deeper than the other. This is typical. We're not that good at getting them to go where we want. And that's just, you have to get down to the sort of sub-gram level to be accurate enough to get them to, be within 50 meters, I would say almost. But we can zoom in on this drifting period, and you can see there's like wiggles here. So the shallower one was wiggling much more than the deeper one. And when we analyze those wiggles using a Fourier transform, we find that actually they match really remarkably well with the Brunt-Väisälä, or the buoyancy frequency, at those two different depths. And so this was minion 2 and 3. These were literally the second and the third minions that we deployed. And we were like, man, I wonder if this is just, you learn in like physical oceanography 101 that this is like something, a cork bobbing up and down at an interface. Is that what's actually happening? And I think the answer is no, they actually mostly follow internal waves, and there's a lot of internal wave energy at the buoyancy frequency. And so we've now had the opportunity to analyze a whole bunch of other minions and look at their temperature. And, if it was just bobbing up and down past an isotherm, for example, then you would see a correlation between temperature and pressure at these high frequencies. And we really, really don't. We see interesting stuff in the temperature, but it's much more chaotic and I think may reflect sort of density compensated filaments and other measures potentially of turbulence. And so another thing that we can do with this data by analyzing the temperature and pressure simultaneously is that we can take the local gradient of the temperature based on a CTD cast when we deployed, and then we can reconstruct what the displacement of the minion is relative to the sort of central isotherm that it's landed at, and compare that with the actual depth. And what you see here, these are two minions that were deployed at the same time. So they have the same CTD cast. One of them went 240 meters, one of them went 140 meters. And you can see that they were oscillating with what's actually an internal tide frequency, as well as lots of internal wave motions. And the actual displacement relative to the isotherms is much smaller. It's not exactly perfect, and that's because the minion is a little bit less compressible than seawater, and it has to be by design. If it was more compressible than seawater, it would be like, your dive suit. It would just keep compressing and compressing and compressing, and it would make you more and more buoyant, or less and less buoyant the deeper that you got, so it would eventually hit the bottom. So we can't have a minion that was more compressible than seawater. But we're very close. And we can also do some nice numerical simulations to convince ourselves that the data that we're seeing from the minion makes sense, because we know so much about its compressibility, its thermal expansion coefficient. We can infer a lot about the drag by analyzing some of its behaviors. We are able to build these models that can basically validate that, in some cases, we do a really good job. This is like the red line here is the minion following an isopycnal. And in other cases, particularly where it's very unstratified, we may not do such a good job. Why do we care about this? I mean, I think that I can think of a lot of physical oceanographic questions that could be looked at from this, but one of our, and I'm super excited about that, but one of the core things that motivated us to even start looking at it from the beginning was a desire to actually directly measure sinking rate, sort of the way that we had originally envisioned the Minion to be, with a side-looking camera rather than with a upward-looking camera. So now we have built a what we call the side-eye Minion right there, which has a camera looking outwards. And we haven't yet, we're going to do our first deployments this July, but you know, our tank tests with it drifting in a tank with a bunch of added marine snow are pretty promising. The last sort of challenge, engineering challenge that we needed to solve was like, well, lots of things, right? They profile so they can come to the surface, they can report data and give you GPS location and then go back down, but the Minions don't do that. And so if we wanted to extend our deployments from, you know, what we've been doing recently, which is kind of days, out to weeks or months, how do we track them while they're drifting underwater? And this is again where we can borrow from Rafos float technology. So this is a photo of my next door office mate, Tom Rossby, who's an emeritus professor at URI, holding a Rafos float from 1986. And we basically use the same principle as the Rafos floats for underwater geopositioning, which is we have 3 sources at least, or 2 minimum sources. We have a receiver, an acoustic receiver on board that is specifically looking for the sweep that those sources produce. And all it does is just correlate and say, okay, what's the scale of what I'm hearing? Okay, is it good? What time is it? And so they can reconstruct or we can triangulate the position of the target or the Minion float based on the arrival time of these different sources. And we've developed what's called a roam tag, which is a teeny tiny acoustic recorder. So it's only a few centimeters inside. This is like fully self-contained. We've attached it onto a glider to verify the positional accuracy. So this is the arrival time from one of the sources from the roam tag, and this is derived from GPS on the glider, and see that it's pretty accurate over sort of 100-kilometer ranges to plus or minus about 2 kilometers. And this is really exciting because for those of you who are animal biologists out there, this was designed to really be used to track animals underwater as an environmental tag. And so it could be used on like lobster, like the crab carapaces before they molt, or other pelagic fishes, anything that doesn't routinely come up to the surface. It could be a potential for this type of application. And this is an example of like a Rafos study that sort of demonstrates the density of trajectories that you can potentially achieve, and the spatial range over which you can observe, which is really remarkably large. The reason for this is because we take advantage of what's called the SOFAR channel in the ocean, which is a sound channel that basically allows sound to very efficiently propagate over long distances. And so we can get coverage of thousands of kilometers. And so we have this sort of vision coming together of like lots of Minion floats. Sorry that the words are off the screen, but it just says like, what if we can't always get these floats back? If they've drifted, too far away from the ship, or the ship has other priorities and it doesn't want to go chase down Minions, or, it's just too expensive to do so. And this really led to another chapter in my work that has been a sort of big focus over the last few years, which is making these floats as biodegradable as we can. So I was at a meeting with my friend Allison Santoro, a professor in microbiology at UCSB, and talking about this conundrum. And she was saying, well, you're using this like filament called PLA, common 3D printing filament. It's like bio-based. Like, what if we could add microbes to it and make it eat itself? And I was just so excited about that. And I was like, yes, like we really need to make our Minions like eat themselves at the end of their life. And we, we kind of thought about it. We didn't really do much for a couple years because we didn't know how to make that happen. But then under this new NSF TIP directorate, there was something called the Convergence Accelerator, which had an emphasis on sort of blue economy relevant stuff. And so we're like, okay, well, if we're ever gonna try to do this, we're just gonna try now. And we got some feedback from the program manager immediately that was like, no, we're definitely not going to fund you to solve your problem, making your robots biodegradable. You have to prove that there's like more of a market and, a broader reach. And so we added a couple other members to our team. Anne is a living materials and synthetic biologist, and Allison Pija is at a company called Mango Materials, does bioplastics. And we basically sort of stepped back a little bit and just started to assess the idea of, adding microbes and other things to bioplastics, specifically for the marine environment, with a goal of making the degradation triggerable and tunable. And that's what gave rise to our little startup company called Myriad Biomaterials. And, through that, we use a biopolymer called polyhydroxyalkanoates, which are made by microbes as a food supply and therefore very easily and rapidly broken down by microbes, including microbes that live in the ocean. We started off just by putting this material into the coastal zone, looking at what microbes populated it and what were eating it really effectively, and then looked at what else they might be missing in their diet. And that led to sort of non-living material, additives, nutrients, things like that. But we also started to work with microbes that could be added directly and survive the process of, high temperatures when it comes to forming plastics. And so we have experimented with two different approaches. One of them is 3D printing with living materials and PHB, and we built the first dual head living material and PHB printer in my lab. So now we can sort of print layers of living material and PHAs. And then also embedding the microbes directly through sort of injection molding processes. That's sort of the vast majority of a lot of the plastics that we use today are through formed by injection molding. And we've had like really good success in getting a lot of these cells to survive that process. It's actually a pretty short process typically. And so once they're embedded in the cells, they seem to have sort of be fairly stable and have a pretty long shelf life. And when they're immersed in seawater, they sort of wake up and then start degrading the material. So these are a bunch of discs that we've made with different sort of formulations of the microbes embedded. And what we're getting is like, a much faster degradation rate than other plastics in the marine environment. And now we're able to make our minion internals fully with that microbe embedded filament, but we're hoping to sort of also work with other potential stakeholders, other ocean instrument manufacturers, aquaculture and fishing, ecosystem restoration, and government and regulators. And really, I'm hoping that it can contribute to like what I see as a growing sort of ocean of things that, could benefit from having understanding and maybe added, increased adoption of biodegradable materials within them. And that's my presentation. I wanted to leave you with a video of the minion that my daughter narrates, and we made together during COVID. So she was 5 years old. And hopefully the sound will come through. I'll turn my volume all the way up.
Hello, everybody. So this is my mommy's minion, and it gets dropped from a ship into water, deeper and deeper and deeper. It goes and goes, and then it gets deeper
Sorry, Zoom folks, we got a little mess up, so I'm gonna start it again. Let's see. Oops.
Hello, everybody. So this is my mommy's minion, and it gets dropped from a ship into water. Deeper and deeper and deeper it goes and goes, and then it gets deeper, and then it stops before it reaches the bottom, and then it takes the pictures, and then it drops the weight, floats back up, and then it goes back up, up, and then there's the data coming from that thing.
It's a little bit, there are other, I mean, like everybody's program, there's lots of other facets, but yeah, I didn't get to talk about the Ghana stuff. Oh, I see. I know. My family loves that. And they're like, she explains your work better than you do, for sure. Yeah. Yeah.
Thanks. Yeah, that is great. Thank you very much. I understand if anybody has to leave, but I think we have some time to take a few questions.
Mhm.
So, questions? Comments?
Thank you for your comments. So maybe if you could share with us how you conceptualize the starburst idea with the weight station. Like, is it a real starburst you can?
Yeah, it's actually a lifesaver.
Maybe you repeat just so it's phrase
Oh,
to phrase.
sure. Yeah. So the question is to talk a little bit more about how the candy ballast drop works. Yeah, so it's a lifesaver candy, which is a ring shape, right? And so you take a little piece of monofilament that has like is basically neutrally buoyant, so you can use like a variable amount of. We have to be so careful about how much when we add weight to the minions. So you take the monofilament, you tie it on, and then you tie another piece of monofilament on the other side of the lifesaver candy, and then you tie a little bit of weight. So typically we use like a nut, like a stainless steel nut or something like that. Maybe about 20 grams. And as the minion falls in seawater, the candy dissolves, and then eventually it breaks because of the weight of that nut, and then that falls to the bottom. So now the minion is 20 grams lighter than it was. And the reason why we have to do that is because at first there are like little air bubbles and things are just a bit more compressible than they would be once you've got something squeezed. It's a little bit like if you're on like a research boat and you need to like submerge the CTD to get the bubbles out before you get good readings on your salinity sensor. If you can submerge something, it squeezes the bubbles down and any compressible parts and creates a little bit of adjustment so that the minion then becomes a little bit smaller. And that then makes it so that it doesn't just pop right back up to the surface. Like the reason why I showed Minion 2 and 3 is because Minion 1, we were so nervous about putting this lifesaver with this clearly death weight attached to it that we were like, we're not going to do it after all. We knew that we could and that we should, but we had never tried it in the ocean. We're like, no, we're not going to do it. And the Minion just didn't sink. It needed to have that extra weight to be able to get down.
Great talk. I'm wondering about some of the particle size and resolution of the SNOCAM from the great talk, and I'm curious whether like that would be sort of the resolve and that be able to like distinguish smaller particles. So it's creatures.
Yeah, That is a like sort of universal challenge with cameras, right? Is that you have to sort of choose what your field of view looks like, and then what your sort of focal depth is, and that affects like the level of magnification essentially that you choose. And so there's always a trade-off between the area that you can view and the resolution. And so in our case, because we're interested in marine snow, and marine snow that leans a little bit towards the bigger side of the spectrum of ocean particles, then we chose something that has a pixel resolution of about 40 micron or so, maybe 20 micron. But in terms of the objects that we can resolve, that means that it's more like 200 micron size, and then up. The crabs are kind of vicarious. We've also had like squid that just grab onto it and other things. We don't know when that's going to happen, and it's always really cool, but it's not quite what the Minion was designed for, although I'd love to consider ways in which it could be adapted to look at other kinds of creatures in the ocean. So I would say that we are constrained on the small end, but not like only in so much as that was our choice. If we wanted to get something that was even more magnifying, we totally could. You would just have to trade off on the field of view that you have, so you wouldn't be able to see all the big things.
Can I jump in on that one? Do you have a library of images so you can actually identify and count?
Yes, we're getting there. We're developing a library of images, and then what you can do with that is you can train machine learning models to then be able to basically process the data on board the Minion. And that's where we're really trying to move toward, is to be able to do that processing on board so that we don't have to send back full images. Like one image right now costs $1,000 in Iridium costs. So it's like equal to the cost just to get one full resolution picture, which we wouldn't do, of course. We'd do some sort of compression, but just as a sort of context.
What's your long-term battery deployment time or goal?
Yeah.
And then over long periods of time, is the mass of the settling snow enough to change your density?
The settling snow question is a really good one. Generally, I don't think so, because it's almost neutrally buoyant, but we could definitely do a back of the envelope and constrain that. The other thing that's kind of remarkable is like the animals that grab onto it are also basically neutrally buoyant. Like we had this squid land on it and hang out for a while, and we're like, oh man, we're totally going to see this in the pressure record. And it kind of goes down a little bit, and it like maybe seems like it's behaving a bit differently, but so much stuff that lives in the upper ocean is neutrally buoyant by necessity. So yeah, I think that it's an interesting question to pose, but it's probably unlikely to affect our ballast too much. We might be able to model it and account for it. And the other question about battery life, I think that it's entirely a function of what you choose in terms of your duty cycle. So I envision these at first to be in this like very slowly changing environment that you'd only need to take a picture once every couple of hours. And that's probably true if everything was perfect. But the reality is that things are changing all the time. Like the higher resolution that we observe with our photos, the more variability we see. And so as we've been doing these short deployments, we're like, well, we're just going for broke and taking as many pictures as we can and measuring pressure and temperature continuously so that we get all that data to be able to interpret what's going on. If we ran it continuously, it would only last for about 3 days. If we duty cycle it, like I did for the SnowCam exports, where it was going every 4 hours, it would last for 2 months.
There could be a lot of interesting applications for these biodegradable biomaterials, and I was just wondering how long do they last in the ocean before they start?
Yeah, so it's very, very much an order one function of the surface area to the volume. So, you know, that's something like this is made out of our material. This would take about probably 6 or 7 years to fully biodegrade. You know, with enhancements, we could like, you know, probably cut that down by like 3 years or something, but
Creating a lot
Yeah.
of surface area.
yeah, creating a lot of surface area, and it's a little non-linear too. So, but yeah, order one, it's just that. I think that, yeah, our goal with the living material additives and the, you know, just the idea, like, can it be triggered? Can it be tuned? How much can we understand about the mechanisms of biodegradation of biopolymers in the ocean, and what are the implications for that? Those have all emerged as really interesting sort of questions that have come out of this work. Yeah, and we've,
Triggering the idea.
Yeah. yeah. So the simple example of that is like my minion floats, where the internals are made out of this biodegradable material, and you would have a purge at the end of life that would introduce seawater, and then the degradation would start. So that's in other applications, like if you just put this in the ocean, there, like, we don't currently have a way, if it's all made out of the same material with the same like you know, homogeneous material, then we don't have a way to trigger it. But if you had it protected in some other fashion, where you could imagine like a layered thing, or even just using the cells themselves to act as a protection for the degradation process. So using spores instead of living cells, and we've tried both.
Cool. So earlier on, you had the, after you introduced the snow can, you had the experiment, was it Explore or something like that, where you found the baseline and the spikes.
Yes,
When you go back to that slide,
sure.
maybe you said it, but I didn't catch it. What triggered that event?
Yeah, that's a great question. The sort of, it's still kind of getting
Yeah.
hypothesized,
Okay.
but there's a few things, right? So this is the classic North Atlantic spring bloom scenario, where you have you have a very deep mix layer, you know, 200 meters earlier in the winter, and then you have restratification. That restratification can trap phytoplankton near the surface, where it, you know, it still has lots of nutrients left over from the deep winter mixing in the winter. The days start to become longer in the spring, so you have more time for photosynthesis. You get a big spring bloom. The bloom starts to run out of nutrients. That might trigger aggregation and then a sinking flux. There's other hypotheses about zooplankton, like Sverdrup's critical depth hypothesis, that say that the zooplankton take a certain amount of time to sort of reach or that the mixed layer can trap them, so that then there's a sort of opportunity to begin grazing the phytoplankton down, such that you see a decline. Here, I think there's also hypotheses coming out of the exports literature from this campaign about a series of storms that occurred during this time. So it's actually very, very rough conditions that we encountered, and they're sort of developing ideas about how the turbulence in the upper water column can, in some cases, act to enhance aggregation. In other cases, it can disaggregate, so it's a little bit of an interesting sort of thresholdy process, but there's some theory that the enhanced turbulence in the upper ocean associated with the storms might have acted to enhance aggregation, that then led to larger sized aggregates, which then, according to Stokes velocity, will sink faster than small aggregates. So you can sort of reach a point where you start sinking out more quickly. Yeah.
Cool. That's what I thought.
Yeah,
Okay.
yeah.
Any more questions? Thank you very much, Melissa.
Thank you.